DNA Monomers, Polymers, and Nucleic Acids by Sadia Mohammed and Sibu Varghese Jacob. What is the difference between DNA, deoxyribonucleic acid, and RNA, ribonucleic acid?
There are four main differences between DNA and RNA. The first is that DNA is a double helix, while RNA is a single helix. The second is that instead of the nitrogenous base thymine, RNA has the nitrogenous base uracil. And instead of deoxyribosugar, which DNA has, RNA has ribosugar. The difference between those two is that there is no oxygen on the second carbon of the deoxyribosugar, while there is one on the second carbon of the ribosugar. The fourth difference between DNA and RNA is that DNA cannot leave the nucleus of a cell, but RNA can.
What are the four nitrogenous bases in DNA?
There are four possible DNA nitrogenous bases, and they can be separated into purines and pyrimidines. Adenine and guanine are both double-ring structures, and they're both purines. Thymine and cytosine are both single-ring structures, and they're both pyrimidines.
How are DNA monomers made?
A DNA monomer consists of a phosphate group attached to a deoxyribose sugar attached to a nitrogenous base. There are four possible DNA monomers because there are four possible DNA nitrogenous bases. There is an adenine one, a thymine monomer, guanine monomer, and a cytosine monomer. To make the monomer, you have to perform two dehydration reactions. The first attaches the phosphate to the deoxyribose sugar. Then you take that and you attach it to any of the four nitrogenous bases. Both reactions produce water.
How do you make DNA polymers out of monomers?
In order to make DNA polymers out of monomers, you have to attach the monomers together with a dehydration reaction. After that, you would have the deoxyribose sugar of one monomer attached to the phosphate group of another. Then you have a single strand of DNA, but DNA is double-stranded. So you have to attach that single strand to the appropriate base pair. A attaches to T with a double hydrogen bond. G attaches to C with a triple hydrogen bond. And of course, A stands for adenine, T stands for thymine, and G stands for guanine, and C stands for cytosine.
And that's a DNA polymer.
There are 6 basic types of mutations. The first is insertion, which is the addition of one or more nucleotides. Deletion is the removal of one or more nucleotides. Substitution is swapping one nucleotide for another. A missense mutation results in an amino acid being changed. A silent mutation is one that does not result in a change in the amino acid sequence. A nonsense mutation results in a stop codon, and it is often lethal to the cell. Mutations aren't always bad. A series of mutations is basically what evolution is.
How do you make chromosomes out of double-stranded DNA polymer?
After you have double-stranded DNA, you can form chromosomes. To do that, first you have to have the DNA wrap around itself. Then it wraps around histones. Then the histones wrap around themselves to form chromatin. Then the chromatin wraps in a spiral to form chromosomes. And there are 24 chromosomes in the average human genome, including the chromosomes that define your gender.
Disorders involving chromosomes.
Some disorders involving chromosomes are Klinefelter syndrome and Down syndrome. In Klinefelter syndrome, you have an extra X chromosome in males. It occurs in approximately 1 in 1,000 males, and symptoms include sparse body hair, breast growth, and wide hips. With Down syndrome, you have a short stature, behaviors associated with ADHD, and heart defects.